In this video, we will discuss about the roles of glycans in the immune response and how these properties of the glycans can be used for therapeutic approaches. So, the roles that we will talk about in terms of the immune response when the cancerous cells overexpress certain type of glycans. So, in this video. The figure on the left hand side describes how the aberrant glycosylations that are expressed on the cancerous cells help in the immune suppression or immune evasion, whereas the figure on the right hand side will discuss about how these glycan roles can be exploited for the therapeutic approaches. So we will start with the figure on the left hand side where the aberrant glycosylation has been shown on the cancerous cells, and I have discussed about this aberrant glycosylation many times in many of our previous videos that most of the solid tumors has this kind of aberrant glycosylation, and in the case of pancreatic tumor cells, these TN and STN antigens glycosylation has been observed more commonly to be overexpressed, and these are the results of truncated O glycans, as this TN is the first step towards O glycosylation and the STN is the second step towards O glycosylation. So these TN antigens will interact with these macrophages and dendritic cells, or these MGL receptors that are present on the macrophages and dendritic cells, and that will trigger anti-inflammation response and angiogenesis. Angiogenesis is important for the cancerous cells because of their inherent properties of uncontrolled growth rate. They need a continuous supply of the food, and even though they reprogram themselves in a way that most of the energy is utilized for their uncontrolled, most of the energies are being utilized for maintaining their tumor properties, but at the same time they also need the additional supply of the food, and these supplies can be met by these new blood vessels that can be formed through this angiogenesis process. So in this term, the angiogenesis is important to maintain the food supply for the cancerous cells. So apart from this TN antigen, the STN antigens are also important for the survival of the cancerous cells, as the sialic acids that are present on the STN antigens interact with the Siglec receptors that are present on the immune cells. In this figure, it is mentioned only about the NK cells and the function that will get activated when the Siglec receptors that are present on the NK cells interact with the sialic acids on the cancerous cells. There are either STN antigens, or the sialic acids are also expressed on these GD2 antigens. So in both the cases, it will trigger the similar response, that is ITIM mediated cascade that triggers anti-inflammations, and this will lead to the immunosuppressions. So these are some of the examples how this overexpression of these glycans on the cancerous cells will lead to the immunosuppression, at the same time this angiogenesis. So how these informations can be used to develop therapeutic approaches. So one of the approach that can be used is that one of the approaches that can be used is to prevent these T cells. So in this example, the MUC1 secreted MUC1 will interact with the T cells and leads to this deactivation, and this thing can be avoided, and this thing can be easily avoided by making these T cells receptors already attached to this MUC already attached to this MUC1 or CA15-3, and thereby this MUC1 that are expressed by the cancerous cells will not be able to interact with this T cells, and they will not be able to deactivate the T cells and reduce the proliferation. So this is one of the approach that can be used to prevent the immunosuppressions. The other things that can be used is about this MUC16. One of the approach that I have currently shown is already mentioned here, that is anti-MUC16 therapy, and this is the problem. The A part is the problem that can be arrived, and B part is the solutions. So in the A part, the overexpression of the MUC1 will lead to the deactivation of the T cells and also reduce the proliferations as a consequences of deactivation of T cells. And in the B part, what is shown as how this can be avoided. So first one is this anti-MUC16 therapy, that is T cells anti-MUC16 therapies can be used, that is uh, this, and this MUC16 therapies can be used using different approaches, and three approaches are shown here, that is CAR T cells, monoclonal antibodies, and T cells will bite. So what the function of these antibodies is to shed this MUC16 that are expressed on the cancerous cells, so that these cancerous cells are being devoid of the glycans, are devoid of this overexpression of the glycans, and thus will not lead to the immunosuppression, thus will not interact with the immune cells, and thereby will not lead to the immunosuppressions or angiogenesis formations. In the case of this bite, there are, apart from the receptors for this MUC16, it will also has the antibodies against another receptors that will trigger the immune response or anti-tumorigenic response. And this anti-MUC16, it will mainly target on this CA125 region of this MUC16, that is a unique epitope for this. That is a unique epitope for all the mucins, that is either in mucin 16 or in mucin 1. The CA mucin 1 and mucin 16 are different. However, the common epitope, the unique epitopes that are found in this MUC16 is this common and no, not common. The unique epitope that are found in MUC16 are this CA125 that can be targeted using this CAR T cell therapy or monoclonal antibody therapies, and thereby that helps in the shedding of this MUC16 from the cancerous surface, and thus it will prevent interacting with the the interaction with the immune cells towards immunosuppression will be reduced, and immune cells can work against the cancerous cells. So this is how the glycan uh, roles in the immune response in case of cancerous cells can be used for its therapeutic approaches. So I hope you got a basic idea how these glycans can be used. Glycans roles can be used for developing the therapeutic approaches in the case of solid tumors. If you need to understand more about these approaches, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.